So here we are in FSN 328. Lipid lecture 4, last one. Lipid lecture 3 is part of that assignment, right? Any problems on that? I apologize, a couple of you had problems with that. It should either run with QuickTime or VLC. Doesn't do that, let me know. It did. Hopefully it should. We're going to do lipid lecture 4. Today's about basically oxidizing lipids, basically burning fat, and then the last part of the middle part of this is going to be what happens when you burn significant amounts of fat in the context of low carbohydrates. So we'll talk about ketogenesis, and then the last part of this will be how to make fat. I'm an expert, so I'm good at talking on this. As you get older, you become more of an expert on making fat. Look forward to that. So this is what we're going to go through. Okay, so this enzyme is kind of the focus of the first few slides here. This is called hormone-sensitive lipase. And as its name implies, it is regulated by hormones. Okay? Now, let's just kind of take a step back, look at big picture and say, when would you expect fat oxidation to go up? So in other words, we have two fuel sources that we primarily use, right? And what are those?
Carbohydrates.
Carbohydrates, specifically glucose and triglycerides, fatty acids from these. I've said a few times in here, in general, the preferred energy source, glucose. So one of the things that affects fat oxidation and how much fat we're going to burn is the presence or lack of glucose. So right away you should say, well, if this is regulated by hormones, insulin must play a role in this. What other hormones? Glucagon. Any others? Epinephrine. When you do physical activity, that can promote fat oxidation. So keep that in mind, and this is kind of how it works. Here's an adipocyte. So here's a fat cell. And these are in lots of different locations in our body, mostly in adipose tissue. That's an accumulation of adipocytes. Now, adipocytes' main function, store triglyceride. So they'll go kind of through different stages of morphology where sometimes an adipocyte will have 80% or something like that of its volume, triglycerides. It'll actually push other organelles out of the way so it can store more triglycerides. So here's a triglyceride droplet. Here, this little black dot is hormone-sensitive lipase. It kind of hangs out in the cytosol until it's activated. And if you look at this, guess how it's activated? It's phosphorylated. Good. It's an enzyme that can be phosphorylated to activate it. Now look what it does when it's activated. It moves from the cytosol to the surface of the triglyceride droplet. Once that occurs, look what's happening down here. What's FFA stand for? Free fatty acids, exactly. So the triglycerides are being hydrolyzed to free fatty acids. Now, these molecules will start to accumulate in the adipocyte, and then they'll run down their concentration gradient out of the cell. That's all that's happening there. So the free fatty acids go down their concentration gradient out of the fat cell into the blood. And here's one of our favorite proteins, albumin. Remember, albumin has those hydrophobic pockets, and it's a good spot for these free fatty acids because they're hydrophobic. So it can bind to albumin, and that'll transport it through tissues. So we'll look at more detail on this in a second. Went through these first 2 points. The last one is it works. Hormone-sensitive lipase works wherever adipocytes are found. Most common tissue is adipose tissue. Hormone-sensitive lipase, though, is also in muscle and a few other tissues. You actually have triglycerides in muscle fibers a little bit. And that can be a source of energy for those under certain conditions. So here's regulation of hormone-sensitive lipase. In the fed state, so in other words, after you eat a meal, if it contains carbohydrate, insulin goes up, right? And that decreases hormone-sensitive lipase activity. I'll show you that in 1 second. During fasting or exercise, Stored triglycerides used as a major energy source. Glucagon and epinephrine activate hormone-sensitive lipase. Therefore, in times of lower blood glucose or exercise, free fatty acids serve as a major energy source, for tissues absorbing them from the bloodstream. So even if, for example, this triglyceride is being stored, let's say right here in the abdominal region, if I'm running or walking or riding my bike and it's my leg muscles that are doing most of the physical activity, those fatty acids can leave adipocytes in my abdominal region, bind albumin, go through the blood, and they'll go into those muscle fibers in the exercising tissue. And that's how they get energy. So when you exercise, you think free fatty acid levels in the blood go up? Definitely. Okay. Free fatty acid levels in the blood tend to go up. So do glucagon and epinephrine. Right? So this slide shows some of that. So let's say this is an adipocyte. So we're looking inside the cell. In the blood, here's epinephrine. Remember, epinephrine is a hormone. It has to have a receptor. So fat cells have receptors for epinephrine, which is good. That means when we exercise and epinephrine goes up, it can activate hormone-sensitive lipase in fat cells. Glucagon, same thing. What's causing glucagon to go up? Drop in blood glucose. In general, when you exercise, blood glucose will tend to drop. It'll tend to drop to about fasting levels. Even if you're consuming carbohydrate, it's hard to get blood glucose to go up. That glucose will tend to run in the muscles, and then glucagon tends to be released. So that's why that's happening. So one of the effects of glucagon is here. It's causing phosphorylation of hormone-sensitive lipase. Oops. This arrow, sorry, I photoshopped this. I hope it's all right on your slides. Hormone-sensitive lipase is being activated here. So now triglycerides forming 3 fatty acids go into the blood, bind albumin. What we're going to do in a minute is we're going to follow this fatty acid. We're going to go all the way through this process to ATP production. What
happens to the glycerol?
It can go up. It can go into a variety of tissues. One of the tissues it can go into is the liver. It's going to be kind of the same thing. It'll start to accumulate in the blood and then start running down its gradient into tissues. Now here's insulin. What do you think insulin should do? Increase or decrease activity of this enzyme? Decreases it. So if insulin goes up, it inactivates hormone-sensitive lipase by dephosphorylating it. During exercise, would you expect insulin to be higher or lower? Tends to be lower. Now, what about post-exercise? I've said a few times in here, after exercise is good to, for your kind of recovery, and a better term for me is restoration from that exercise, is carbohydrate and protein. But here's the issue. If you consume carbohydrate and glucose, what's going to happen to insulin? It's going to go up. And what will that do to hormone-sensitive lipase activity? Decrease it. Okay? Is that a problem? Depends on your goals, right? I'd say in general, probably not, because at this point, what's happened is, let's say you're done exercising and now you have your carbohydrate and blood glucose goes up, you'll slow down hormone-sensitive lipase, but a lot of those fatty acids are already in the blood. Those fatty acids will continue to go into tissues and be oxidized even as insulin's going up. Okay? Then what's going to start happening is you're going to start switching over. And if you keep insulin high for a while, have you ever heard this? This is one of the caveats one of the footholds of the people that say carbohydrates are not good, is they'll say Carbohydrates promote fat synthesis. We'll look at that coming up, but overall, the positive effects of that carbohydrate post-exercise are more beneficial than any negative effects from inhibiting hormone-sensitive lipase. So, not an issue there. So let's start following these guys through. How do we use fatty acids for energy? This will be review from biochem, I hope. Those fatty acids can be oxidized to produce FADH2, NADH, and Acetyl CoA. Remember this, per weight, fatty acid oxidation yields more ATP than glucose. We're going to look at that and why that occurs. Beta oxidation is a major pathway of oxidizing fatty acids for energy. Beta oxidation occurs in the mitochondria. Why is it important that it takes place in the mitochondria?
Utilize acetyl CoA.
And? Somebody help Aiden. He got one for you. Yeah, these guys. ETC's in there. So think about what's happening. We're going to oxidize the fatty acids. We get electrons on FAD and NAD, and that's where ETC is. So that's efficient. And then also acetyl CoA can go into Krebs cycle. That's inside the mitochondria as well. So what you're going to do on your poster is follow this process. On the instructions for the poster it says, show how a fatty acid is oxidized. Start with the fatty acid coming in from outside the cell. So you can have the fatty acid going into the cytosol of your cell. Then you're going to activate it. So here's the activation step. After fatty acid enters the cell, it must be activated before it can be oxidized. Activation means adding coenzyme A, and this occurs in the cytosol. It requires 2 ATPs. Why are we doing this? Coenzyme A is essential for fatty acid transfer into the mitochondria. Remember that? Let's talk about that in another class. If not, here it is. Here's the fatty acid. So let's say, for example, it comes in from out of the cell, goes through the plasma membrane. Or let's say this is I'm going to be optimistic and say this is a muscle cell. And I'm hoping it can burn my fat. So here's my fatty acid. This is albumin. All my proteins look alike. You can probably tell at this point. This fatty acid goes in. This is what we're doing right here. Fatty acid combines with coenzyme A. The enzyme that does that is that fatty Acetyl CoA synthetase that we talked about before. Requires 2 ATP. You can see that because we went from adenosine triphosphate to adenosine monophosphate. 2 phosphate equivalents are used. Now we have this guy. Reason we have that is to get it into the mitochondria. Now what we have to do is this. One of the things we've said over and over again is transport through the mitochondrial inner membrane is stringent. It takes coenzyme A and carnitine. You heard of carnitine before? If you haven't, maybe now you'll be kind of tuned into it. A lot of times carnitine is promoted as a fat-burning supplement. Got to have carnitine burn fat. And that's absolutely true. Sometimes what they don't say is we tend to make enough carnitine to burn all of our fat. So we don't need to ingest it. But this is how it works. So if we kind of look in detail, here's the inner membrane of what? Mitochondria. So here's the matrix. Here's the intermembrane space. So essentially we're right here. For that fatty acid to get through the inner membrane, there's an exchange that takes place. Coenzyme A doesn't go through that inner membrane. That's basically why acetyl CoA doesn't leave, right? So coenzyme A comes off. There's an enzyme right here that takes CoA off, replaces it with carnitine. Carnitine is a small peptide. It's a very small molecule made from 3 amino acids. Like I said, our body generally makes as much carnitine as we need, so in general, save your money. You're going to take that. Now you have carnitine and fatty acid. That moves through the inner membrane. Remember, this is a lipid bilayer. Moves through the inner membrane, then look what happens. A second enzyme switches it back. Carnitine comes off, coenzyme A is added. You don't have to remember this, but this might kind of help understand the process. This enzyme here they call CAT1, carnitine acetyltransferase 1. This is CAT2, carnitine acetyltransferase 2. That enzyme is just basically switching carnitine for CoA, both of them are. So what did we do? We got the fatty acid inside the mitochondria, and it's got coenzyme A attached. So now it does this, right? Coenzyme A off. On the inside, we now have CoA. Oops, CoA is added on the inside of the matrix. Why is that important? Because that's the substrate for beta oxidation. Why do they call it coenzyme A? Because it's required by quite a few different enzymes to function. One of the enzymes is in beta oxidation. So here's beta oxidation. And I'll show you the diagram of this in 1 second. Step 1 is the activation of the fatty acid by adding CoA, then transferring it into the mitochondria using carnitine. Step 2 of beta oxidation is an oxidation step. That generates FADH2. Then hydration, so water is added to the molecule, then another oxidation step. So one way to think about beta oxidation is beta is 2. There's 2 oxidation steps. Now we get NADH. And step 5, CoA is added. That's a second coenzyme A. So what does it do? That generates acetyl CoA. Is this bringing back memories? Good memories, I hope. Now, for your poster, you can replicate this. There's another diagram in your text that shows it in kind of a circular or cycle. To me, this one's a little bit easier to follow. And I wrote in the steps from the previous slide. So this is palmitoyl CoA. If you hear that name, what does that remind you of? That's palmitate. That's palmitic acid, coenzyme A added. So how many carbons? Palmitate. 16. That's one of the most common fatty acids in the body. So we're using that as our example. Here's the structure, right? Palmitic acid is saturated. It's a saturated fatty acid. First step, and you don't have to remember this enzyme, any of these enzymes actually. Palmitoyl CoA, palmitate, CoA, is oxidized. And as the name implies, is a dehydrogenase that does it. So hydrogens come off, they're added to FAD. So now you have FADH2, this hydrogen comes off, this hydrogen comes off, added here. Now you have this, you have a double bond. See? See the way there's only one hydrogen here now, one hydrogen here, because they were added to FAD. Why is that important? Because FADH2 can go to ETC. We've already generated 2 ATP. Next step, hydration. So H2O is added to this part of the molecule. OH here, hydrogen here, H2O here. Next step, second oxidation step. This enzyme is a dehydrogenase. This time it's adding the hydrogens to NAD. This hydrogen comes off, this hydrogen comes off, added here, we get NADH. Why is that important? That can go to ETC for 3 more ATP. We've already made 5 ATP. Haven't lost any carbons yet. Next step is when we cleave. So this is when coenzyme A is added, and we obtain acetyl CoA because of that. So to follow this, here's our coenzyme A. Now if you look at this, you guys know this structure. What's that structure look like to the right of my hand? If this is a CH3, what is that? That's acetyl CoA. So what happens is coenzyme A is added right here. That removes an acetyl CoA. So now you have an acetyl CoA here and a fatty acid that's 2 carbons shorter. Why? Because they're now part of acetyl CoA. So 2 carbons come off and form acetyl CoA. That can go to Krebs cycle. How many carbons are left? 14. 1 here, 12 here, that's 13, 14. So now what happens? Repeat it. So don't forget your arrowhead here. It's a little hard to see, that's an arrowhead. You repeat this, repeated sequence.
For the poster, do you want us to use that specific fatty acid?
Yeah, that's probably easier. Yeah, just draw out those structures real quick, because then you can kind of follow. And the reason we're going to do it in that detail is so that you can basically calculate how many ATP are made from each fatty acid. And have you done this before? I'll tell you a shortcut on how to do that in a second.
So this is all in the mitochondria, right? The
Yeah.
matrix. Do you want us to show the steps before, like how it got into the matrix?
Sure.
On our poster?
Yeah, you'll show the fatty acid going in. You'll show the activation step. You can just show the transfer step. You can like enlarge your membrane a little bit, put carnitine, and that'll help you kind of remember it. Carnitine helps it go in. And then you can draw out your beta oxidation.
Are those 2 enzymes, are they like on the membrane, or does that happen in the cytosol and then it's a different molecule when it goes into the membrane?
This enzyme that's activating it,
Yeah.
cytosol.
The cat
There's-
1 and 2.
They're on the mitochondrial inner membrane.
Okay.
Yeah. Let's go through this. So now, repeat it. You're going to repeat this until all of the carbons are part of acetyl CoA. So if you have a C16 fatty acid, how many acetyl CoAs does that generate? 8. C16 divided by 2 carbons is 8. 8 acetyl CoAs. How many cycles do you need to do that? 7. see if I did this right. After the first cycle, acetyl CoA, C14. Do that again, and again, again, again. 1, 2, 3, 4, 5, 6. On your last cycle, you have 4 carbons. 4 carbons coming through. When that splits, you have 2 acetyl CoA. That's the end of it. So put this in your notes, and this will make sense hopefully now and definitely later. The number of cycles for oxidizing a fatty acid with an even number of carbons. The number of cycles equals the number of carbons divided by 2 minus 1. The number of cycles equals the number of carbons divided by 2 minus 1. Make sense? Show you an example in a second. Let's add up the ATP. Here's palmitate. Now remember, each time we go through this, if we call this a cycle, we generate an FADH2 and NADH. That way we can get 2 and 3 ATP for a total of 5. So every cycle we get 5 ATP from FADH2 and NADH. Oops. That's why this says 7 cycles. From these guys, we get 5 ATP every cycle. 7 cycles times 5 is 35 ATP from that. If this is a C16, we can get 8 acetyl CoA. And you guys remember this. Every turn of the Krebs cycle gives us 12 ATP. So 8 acetyl CoA going through Krebs, 96 ATP. Add those up. That's 131. 131 gross ATP. If you do net, you got to subtract the 2 that you had to use to activate it. So this step here required 2. So you subtract that, that gives you 129. So you want to be able to calculate the number of ATP that can be made from saturated fatty acids of different lengths. I'm not going to ask you an unsaturated fatty acid. That'd be more or less. Would an unsaturated one be more or less? Less, because there's less hydrogens. Uh-huh.
If fat is so efficient of ATP, why does our body prefer carbohydrates?
They say it tastes better, but that's debatable. Oxygen. Got to have oxygen to do this. Right? You got to have oxygen to run ETC, got to have oxygen to continue to run Krebs. So carbohydrates are the only place we can get ATP, anaerobically that, and creatine phosphate.
Would it also be mitochondria?
Yeah, that's another difference, yeah. One of the things we're going to talk about in 1 minute is they tend to work together. A lot of people don't realize this, but carbohydrates helps burn fat. I'll show you how in a second. Now, you can imagine when you're going through this, you make a lot of ATP. You can make a lot of ATP. A lot of it's from acetyl CoA. Now I want to ask you guys something, and keep this in mind. Why do we get more energy from fat than we do carbohydrate? Yeah, you get 9 kcals per gram of fat, 4 kcals per gram of carbohydrate. Why? Kind of. There's 2 main reasons. One is, when you look at that fatty acid, There's no oxygen in its structure. So it has more potential to be oxidized. It doesn't have oxygen. Compared to glucose, right? Glucose has oxygen in its structure. It can't be oxidized as significantly as a fatty acid. So this is what I want you to think about as well. Let's say we have a C18 fatty acid versus glucose. Which would generate more ATP? This guy, right? Why?
More carbons.
Okay, more carbons, that's one reason. Now what? How many carbons here? 18 if my math is right. 18 here. 18 here. Now which can generate more ATP and why? Still seems like fatty acid, right? And one of the reasons is, well, because it can be oxidized more. It has more electrons to run ETC and Krebs cycle. Which one generates more Acetyl CoA? Why? How many Acetyl CoA are you going to get here? Every glucose, how many do you get? Acetyl CoAs? 2. Why? You see where that would be an issue? Bridge step. Remember, you take 3 glucose, you get pyruvates, and then what happens? When you go from pyruvate to acetyl CoA, you lose a carbon. Okay, so that can be a difference there. So basically, this generates more acetyl CoA. They can go through the Krebs cycle. They can generate more energy. So you're going to be able to write that out on the exam as well. So you're going to be able to calculate this. You're going to say, oh, it's C18. I can calculate how many ATP I get from that. You're going to be able to calculate how many ATP you get from this. And then what would that be? Maximum. Right? Do that math. Calculate. I don't remember it off the top of my head. But just based on what we talked about, this is more than that, right? So you want to be able to do those calculations and explain to me. And here's what we're going for. This is an old saying. Have you ever heard this? Fat burns in a carbohydrate fire. Sometimes they'll say fat burns in a carbohydrate flame. What does that mean? What I just said a minute ago. Carbohydrates help burn fat. And this is how. So this is why I'm always discouraged when somebody says, yeah, I just want to burn a lot of fat, so I cut down my carbohydrates. And I feel like giving this lecture. How does carbohydrate help burn fat? Well, the oxidation of fatty acids can produce large amounts of acetyl CoA, right? So let's say inside the mitochondria, we're going through this, and we're oxidizing a fatty acid. We're going through beta oxidation, and we make all these acetyl CoA molecules. Right? We're going to make tons of them. Acetyl CoA has to combine with oxaloacetate in order to enter the Krebs cycle. Oxaloacetate is normally from glycolysis and pyruvate. In other words, if a cell needs more oxaloacetate, it can go from pyruvate to oxaloacetate. I'll show you that reaction in a second. So, if oxaloacetate is low, right, so if we say, well, this can combine with this, right? Forms citrate, goes through the Krebs cycle, right? And a lot of times I think we think of it that way. Oh, okay, that's going to work. But what if we get a ton of these? And let's say this one can do that as well. Combines, forms citrate. But let's say oxaloacetate is limiting. These two can combine to make a ketone. That's called ketogenesis. Remember we talked about that? We said in low carbohydrate conditions, ketogenesis can occur. So that's what we're going to talk about right now. And here's kind of I took this out of a sports nutrition text. And it kind of shows that process. Again, not a perfect diagram, but it's fine for us. Here's the liver. Does the liver burn fat? Yep. Burns quite a bit, actually. It'll burn fat. So beta oxidation occurs in the liver. So here, for example, is beta oxidation making acetyl CoA. If glycolysis is also running in the liver, remember I said a few minutes ago, fat oxidation and carbohydrate metabolism are typically occurring simultaneously. Like right now, you're getting probably 70% of your energy from burning fat. The other 30% is almost all coming from carbohydrate. It occurs together, and they tend to kind of go back and forth on which one is contributing the most to energy. So here's glycolysis. Now, a lot of times we look at glycolysis this way. Pyruvate to acetyl CoA. What if a cell already has a lot of acetyl CoA? Acetyl CoA will actually inhibit this. So now what does the cell do? It can take pyruvate, go to oxaloacetate. Why is that important? Because now if you have a bunch of acetyl CoA from fat oxidation, that carbohydrate can support that by providing more oxaloacetate. That can run Krebs cycle. That helps us produce a lot of energy from the combination of fat and carbohydrate. What if a person is limiting their carbohydrate intake to the point that carbohydrate becomes limiting? What if they're well below the RDA? What if they're on a low carbohydrate diet? A lot of them will say, we want to curb your carbohydrate appetite. We want to teach you to stay away from carbohydrates. Consume 20 grams of carbohydrate a day for 2 weeks. What happens? Carbohydrate becomes limiting. Liver cells can't go from pyruvate to oxaloacetate. If they do, that oxaloacetate doesn't go this way, it goes this way. Why? What's this? Gluconeogenesis. Why is the liver doing that? Because other tissues need glucose. So now what happens? Acetyl CoA accumulates. So in conditions of low carbohydrate, oxaloacetate can become limiting. That can cause acetyl CoA molecules to combine with each other. And eventually make ketone bodies.
Is it fair to say that any cell that possesses a mitochondria has the ability to do fatty acid beta oxidation? Or are there certain cells that lack those receptors in their membrane?
They have mitochondria. I'm trying to think of an exception. I believe that they... brain cells come to mind. Fatty acids aren't going to tend to go through the blood-brain barrier, so they're going to tend to run carbohydrate through aerobic metabolism, not really fatty acids. Most of the other tissues, that's the only exception I can think of. Most of the other tissues, yeah, they'll run fatty acids through beta oxidation.
I heard about athletes who are endurance trainers, so they go on really low carbohydrate diets, but then up their fat a lot, so that they basically train their bodies to switch over to anaerobic, or
to aerobic.
to aerobic, like So like bring that level up to here so that they can do that more often to get their energy from that. If that makes sense.
The last time I saw that data or looked through some of those papers and kind of got the consensus was no significant improvement in performance.
Really? Okay.
Yeah. Sometimes they can still train through that. Their body adapts to utilizing fat.
Yeah.
Doesn't improve their performance.
Okay. So you can't actually raise your levels of aerobic.
Sure, you can to a certain point, but can that, now if you take that same group and you say, okay, train aerobically, get your systems up, if now we're going to give you a carbohydrate and lipid diet versus a group that went through the same thing, we're just going to give you high lipids, that high lipids doesn't improve performance.
Okay.
So one of the points is, why would you do that?
Okay.
Yeah. No, that's the last one. That's been a couple of years since I looked through all that, probably. Uh-huh.
Going back to diet reaction what was the benefit?
This being inhibited? I'm trying to remember what I was driving at there. Because then, rather than pyruvate becoming acetyl CoA, if you already have a lot of acetyl CoA, you don't need to do that. It's better if pyruvate converts to oxaloacetate. So if this accumulates, the cell will try to slow down more acetyl CoA from pyruvate. Pyruvate will go to oxaloacetate. So it's a way of trying to provide oxaloacetate to combine with acetyl CoA. Now, here's again an example of what the liver does. The liver can burn fat, can burn carbohydrate obviously, but it can also make ketones. It'll combine acetyl CoA molecules to make acetyl CoA. That becomes ketone bodies. The liver puts those into the blood so other tissues can use ketones. So the liver makes ketones for other tissues. Why? It's an energy source for those tissues. The brain and muscle cells can run on ketones. The liver doesn't. Sure, the liver generates ketone bodies as an energy source for other tissues, mostly the brain and muscle. Right? It's kind of an alternative energy source instead of what? Glucose. The liver itself doesn't use ketones for energy. And the next slide kind of indicates that. So here's ketogenesis. This is kind of what we just talked about. Here's acetyl CoA combining with another acetyl CoA. When they combine, one CoA comes off and you have acetoacetyl CoA. That's the first one here. Now, acetoacetyl CoA, when the CoA comes off, you have acetoacetate. That's the first ketone. So these three molecules that I have circled in purple are the ketone bodies. Remember we said 130 grams of carbohydrates, the RDA, to minimize the partial replacement of glucose by ketogenesis. So in general, these are the ketones we're talking about. Acetoacetate converts to beta-hydroxybutyrate bidirectionally. Acetoacetate can also become acetone. Look what this says. Acetoacetate can revert back to acetoacetyl CoA and then acetyl CoA in extrahepatic tissues. So what does that mean? These are put into the blood. they can travel to muscle and brain. Those tissues can get acetyl CoA back. And then what do they try to do with that? Krebs cycle.
What happens to acetones if it's not reversible to acetoacetate?
Expired.
Okay.
You ever smelled that? Somebody's breath, if they're starving, let's say they're going through fasting or whatever, or sometimes if a person is diabetic, that's another thing that can happen. Is you can actually, that's a kind of a they say it's almost a fruity flavor. And I actually get that taste in my mouth sometimes if I'm doing something. Not as much now, but it used to be if I was in research experiments and I couldn't stop. I was just going and going and going. Sometimes you can actually kind of feel that. You kind of get that odd taste in your mouth, and you know that you're basically generating ketones. Does our brain run pretty well on ketones? Not bad. It's a good starvation alternative energy source. That's what the body is doing at this point is, if glucose is limiting, our body's trying to switch to an alternative fuel source. Brain runs decent on ketones. Some studies have shown some cognitive function not as good on ketones as glucose, but overall not bad. Same with muscle. But what has to take place? Aerobic metabolism. So what is it? Let's talk about this. What is the brain doing? It's taking acetyl CoA, it's getting these, converting back to acetyl CoA. What does acetyl CoA have to combine with? Acetate. Where's it getting that? The liver's doing gluconeogenesis, trying to provide enough glucose to the brain to support aerobic metabolism using ketones. So the liver's making ketones and glucose to try to support energy production in other tissues.
And it'll get that from amino acids?
Some of it can, yeah, exactly. Yep. So why is our body doing this? One of the reasons is when a person goes through the initial stages of starvation, skeletal muscle loss is significant and rapid. So the person starts wasting. One of the reasons there's rapid weight loss at this point on those diets is skeletal muscles breaking down. Why? Because amino acids are being used to provide substrate for gluconeogenesis. So what happens? Rapid weight loss. The other thing that happens is you're becoming dehydrated. Because as you lose those muscle cells, you're losing glycogen as well, and water's used to store glycogen. So you're losing that hydration as well, so you get rapid weight loss. They say it's curbing your appetite, but a lot of times the point of it is cause weight loss in an individual so they'll stay on the diet. And that's kind of overall not a healthy strategy, but that's kind of why some of them work. Okay? Here are the ketones. Their structures. You don't have to remember structures. You want to remember their names. Are those all ketones? Which one's not a ketone?
Oh wait, no, sorry.
Ketone, ketone, not a ketone. Why did they include it? Because it converts back and forth with acetoacetate. This one's interesting. I mentioned this, but this is called starvation in ketone bodies. Now the two main ketones in the blood, hydroxybutyrate, acetoacetate. Acetone's basically in low levels. The reason is because it slowly converts from acetoacetate. So we have 4 carbons here, 3 carbons here. So CO2 comes off. When it comes off, you get acetone, it doesn't revert. So acetone is basically expired in the breath. So it doesn't tend to accumulate. The most important or relevant ketone in the blood, beta-hydroxybutyrate. After a few days, the ketones can become a significant energy source.
I thought you said on the last slide that one wasn't a ketone.
It's not, but it's included as a ketone body because it converts to acetoacetate. So when we talk about those ketones, that one's included because it can convert to this one. A little odd. Now, what this doesn't show is, even overnight, even overnight, as your blood glucose drops and your hepatic glycogen stores tend to drop, ketone production starts creeping up. Now what happens? Hopefully you get up the next morning, you have a nice breakfast. You guys have oatmeal, right? Whole wheat bread, ketones go like this. Why? Glucose goes up, insulin goes up, ketogenesis down. If you don't eat and you continue to not eat, ketone production is going to continue to go up.
Is the enzymes for ketone production or insulin dependent? Like, are they inhibited by the presence of?
I don't know. I don't know how they're regulated. I don't know if they are regulated by insulin. It could be just substrate flowing through those pathways that changes it. Yeah. Ketosis. We've heard that, right? What does that mean? It means you're on your. Used to be, they'd say, got to get to ketosis if you want to be a successful dieter. That means an abnormal rise in concentration of ketone bodies. Basically, that can happen during starvation, a low carbohydrate diet, or sometimes in diabetes mellitus. Why? Because the glucose isn't going into the cells. So it's not metabolized, it's not providing oxaloacetate, and that can cause this. So in that case, you can't use the carbohydrates that are present. Now, one of the issues here is it can lead to ketoacidosis, which means dropping of the blood pH. So your body has buffering systems that are basically resisting changes in blood pH. Most of the time, they're not overwhelmed, and you can maintain your blood pH within 7.35, 7.45. In some conditions, though, ketoacidosis overwhelms the buffering systems, and it can be harmful, including lethal. So in general, ketoacidosis is not considered, shouldn't be considered a healthy or kind of good scenario. Clear on that? Okay, that's probably the last pro-carbohydrate thing I have to go through. Hopefully we're on board. Now, let's look at the other side of this. We're not starving. We're not making ketones. We're making fat. It's good. So this is how we synthesize fatty acids. So the next couple slides, again, you can put this on your poster. You can use these parts that I have in the slides. They're, I think, a little bit clearer than actually what's in the latest version of the text. And this is fatty acid synthesis. So we know this occurs what part of the cell? Cytosol. Beta oxidation, mitochondria, fatty acid synthesis, cytosol. Opposite pathways, different parts of the cell. That's why they're in different parts of the cell. So this is what happens. Fatty acids can be made from acetyl CoA. Acetyl CoA can be made from carbohydrates, proteins, and fats. We've looked at that a few times. And in general, in humans, the main sites for fatty acid synthesis are the liver and adipose tissue. Acetyl CoA is converted to citrate in the mitochondria. Citrate moves from the mitochondria to the cytosol. It's converted to acetyl CoA. Then the acetyl CoA can be used for fatty acid synthesis. So when we have so much energy that we no longer need Krebs, we can store it as fat. So citrate leaves the mitochondria. So if you want on this or whatever, you could put, this is in the cytosol. Remember that? Like, for example, we said if you have a lot of ATP, Krebs cycle slows down. Citrate will actually accumulate, leave the matrix, go into the cytosol. The enzyme citrate lyase splits it back to what it was made from. Now what do you have? Acetyl CoA in the cytosol. That's substrate for fatty acid synthesis. If acetyl CoA is in the matrix of the mitochondria, what is it? Krebs cycle substrate. If acetyl CoA is in the cytosol, fatty acid synthesis. This looks worse than it is, I promise. This set of reactions you don't have to remember, but I'll go through it really quickly. This is how a fatty acid is synthesized. So here's the steps, including the first one. Acetyl CoA can be converted to malonyl CoA. The enzyme that does this is acetyl CoA carboxylase. That enzyme, basically, it's a carboxylase, takes CO2, adds it to acetyl CoA. That forms a 3-carbon malonyl CoA. Over here, see these little shapes here, CE, ACP? These little shapes here represent binding sites on fatty acid synthase. So you can put on your poster in here my instructions for the poster say identify fatty acid synthase. So you can basically do this. Those are binding sites on the enzyme complex. So here's malonyl CoA. It's bound right here. Malonyl CoA has 3 carbons. What's this? What's that look like? Acetyl CoA. So acetyl CoA and malonyl CoA bind fatty acid synthase. Then they're combined. So you go from 5 carbons, they're combined as the CO2 comes off. So acetyl CoA basically slides right here and combines. So now you have 4 carbons. Okay, start with 5, now you have 4. Next step, reduction. NADPH reduces this. Okay, so 2 hydrogens. So this is NADPH2. It's hard to see, isn't it? It's impossible to see because it's not there. Hydrogens added here, hydrogens added here. So reduction. Dehydration. Reduction. Then, this fatty acid moves from this site to this site. Now it's right here. See what it just slid up? Why did it do that? Because this is the site where malonyl CoA binds, this ACP. So another malonyl CoA molecule comes in. Then we kind of start repeating. This slides down. Now how many carbons do we have? 1, 2, 3, 4, 5, 6, because CO2 comes off. So we start with 5, then we get to 4. Rearrange, add another malonyl CoA, now we have 6. Now, repeat. This is an arrowhead right here. These sequences repeat until you get palmitate. So it says right here, cycle repeats until palmitate is formed. Alright, so the first one you get C6. Next one you'll have C8. Then C10, you'll go all the way to C16. And then palmitate's released. So the end product of fatty acid synthase is palmitate. And then palmitate can be added to glycerol to form a triglyceride. So that triglyceride could be stored in adipocytes, or if this occurred in the liver, the triglyceride can be exported in VLDL. That's how the liver makes it. Goes through this. Now,
Do you want us to be able to draw this?
no. For your poster, draw this. That'll take you 5 minutes. But on the exam, I won't make you draw that, but I'll ask you some of these basic questions. One of the questions I'm going to ask you is, how's this regulated? One of the hormones that regulates this, insulin. If you were to guess what insulin does to fatty acid synthase activity, would you say increase or decrease? Increase. Insulin increases fatty acid synthesis by upregulating acetyl CoA carboxylase. Insulin increases fatty acid synthesis by increasing acetyl CoA carboxylase activity, right here. I'll tell you how that works in a second. Another molecule that increases acetyl CoA carboxylase activity, citrate. Why regulate that enzyme? Because that's where the carbons are coming from for the fatty acid. Carbons are coming from malonyl-CoA. You add a malonyl-CoA here and here. So most of the carbons come from malonyl-CoA. The only carbons that come from acetyl-CoA are in the first cycle. From then on, it's malonyl-CoA. So if you regulate malonyl-CoA amounts, you can regulate fatty acid synthesis. We know why insulin would do that because it's an anabolic hormone. What about citrate?
20 Krebs molecules around.
Yeah, that's so when citrate starts going up, that means we already have high levels of energy in the cell. So store some of that as fatty acids for later. Okay. Questions on this? Okay. Let's start protein. If I can find it. Now, one of the things that we'll get right away from protein is it's a lot different. We spend a lot less time on it because it's not an energy source in general. So we can go through this pretty quickly. Here's the outline. I'm thinking I have my notes for protein there. There we go. Why do we need to eat protein? Amino acids, more specifically, the essential amino acids. So we consume dietary protein because it's a source of essential amino acids. Right, we eat lots of different amino acids. How many amino acids are used during translation to synthesize proteins? 20. Remember that? We need 20 different amino acids to translate proteins. How many of these amino acids are essential to the diet? 9. Remember that? 9 of the 20. Here are the essential amino acids. This is for humans. This is species specific. So does that mean essential? Do we make these?
Not adequate.
Essential amino acids are those that we either do not make or make in adequate amounts. We have to get some from the diet. It's actually only about 2 of these that are not made in the body. The other 7 aren't made in adequate amounts. So here they are. You want to remember these. This would be like multiple choice. Is glycine an essential amino acid? Nope. right? I think there's an easy way to remember this. Remember this? Private Tim Hall. Can use a little mnemonic. PVT. Private Tim Hall. Probably shouldn't tell you. Just fill in the which one's for which.
That's not any structures.
I'm going to go easy on the structures. Probably just maybe a couple of them, and I'll tell you why coming up. Okay, so we have to consume those. What if we don't? More specific than we die or you're in big trouble. So you go on Survivor. Keep this in mind. The genetic code determines the protein's amino acid sequence, right? And that is determined by the genetic code. So during translation, what if the genetic code specifies for an essential amino acid, and that essential amino acid is not present? What happens to protein synthesis? Stops. Can't synthesize that protein. Now, if that protein is essential for life, good chance it might be, that's a problem. It's a problem relatively soon, because proteins turn over in our body relatively quickly. So one of the things I would say, if you're on an island, if you're on Survivor, the nutrient you're probably going to fight for first is water. Water. You'll feel the effects of water deprivation faster than anything else. One of the next things I would think of, protein. Because you'll lose that protein relatively quickly. And again, in this case, if you're in starvation mode, remember, your muscles are going to start catabolizing, so you need carbohydrates, but you really need the protein, so keep that in mind. What do these amino acids look like? I'll go through this quickly. We've all seen this. Blah, blah, blah. Central carbon here. Remember, they all have a side chain. They have an acidic group, carboxylic acid group, and amine group, so they all have the same core structure. The difference is just that side chain, that R group. Remember, they're linked together by peptide bonds. The individual that actually worked on this got the Nobel Prize for characterizing peptide bonds. His name is Linus Pauling. Here's one of the amino acids here. Now that peptide bond is formed. Where's a good place for this to be formed in the body? In ribosomes. Ribosomes actually form these peptide bonds. So a ribosome will take two adjacent amino acids, form a covalent bond using carboxylic acid group of one and the amine group of the other. Remember, physiologic pH, these are charged. If I ask you to draw a dipeptide or a tripeptide, charge these. Now when these combine, you lose a water. And a shortcut for drawing these, remember, did you guys draw these in other classes? Okay. So if you draw a peptide, here's the core structure. Just do it like this. There's my first amino acid. Now you can just put in your second one. Now you have to do is kind of fill in. Now I can do the ends. I can say, well, I know this is H3N. I know that's charged. I know this is. You can draw this however you want. Do it this way. Now, here's my central carbon. So I can just put an R on there. What's this? It's a hydrogen. Here's my central carbon here. I can do the same thing. Now all I got to do is this. Well, when you lose a water, that leaves an oxygen here, and this kind of makes it planar. So there's my peptide bond. So NCC, NCC, all the way through your peptide. So even if you don't know the side chains, you know most of it. That's partial credit. So I'll tell you which amino acids to remember next time. There won't be many. Have you guys seen charts like this? Look at all the different things proteins do in the body. I'm not going to go through these. These are pretty self-explanatory. Something that we're going to talk about a little bit differently in here that you might not have other classes is this. How we actually use amino acids for oxidation and gluconeogenesis, and then synthesizing other key molecules. Oh, I forgot to tell you the second reason we consume proteins. Nitrogen. essential amino acids, and nitrogen. So that's what makes it different than the other macronutrients is nitrogen. And we can't, nitrogen is one of the most common elements on the planet. But a lot of times nitrogen is present this way. We can't reduce that. We can't separate it. We rely on other organisms to reduce and separate those nitrogen molecules, incorporate it into amino acids, and then we, on the top of the food chain, consume the proteins, get our nitrogen that way instead. So we have to have it for nitrogen. That way, when we have to make molecules that contain nitrogen, we can use nitrogen from amino acids to make those molecules, like creatine, for example. How much protein is in the body? Protein is about 14 to 17% of the total mass of a lean adult. So, for example, for about 30 pounds of a 200-pound man. Skeletal muscle accounts for 40 to 60% of the body's proteins. Skin and blood each account for about 15%. Why am I saying that? Because if a person needs nitrogen or is breaking down proteins, most of the breakdown, skeletal muscle, because that's where most of the protein is. Uh-huh.
Is the rest found as lipoproteins on plasma membranes, or where's the other 25%?
Oh, for this?
Yeah, so we have 40 to 60 skeletal, 15 skin, but there's still anywhere from 25 to 45%.
Yeah, other tissues. Yeah, skin and blood. Yeah, so it could be other tissues. Pancreas,
Enzymes and
liver. Yeah, overall in those, yeah, that type of compartment. Yeah. Now, storage form of carbohydrate is? Glycogen. Storage form of lipids? Triglycerides. Storage form of protein? There isn't one. It's basically functional. There's not really a storage depot of protein in the body. So we don't store it to use it for later when we need it. We typically make proteins, and if we need nitrogen or something, we break down those proteins. Most of the protein goes in skeletal muscle, so that can be affected. Uh-huh.
For the last slide, did you mean skin and blood together are around 15%, or each of them are 15?
Each are
So
15.
30.
Yep.
That makes sense.
Yeah, so it's pretty close. You ever heard that term, use it or lose it? Yeah, it's sad. Use it or lose it. I have lost so much. Here's what one of the things that means. This has to do with protein turnover. A lot of times you'll say, use that muscle or you'll lose it. Very true. Very quickly. Fortunately. I'm not talking years, I'm talking hours. So if we look at this, that's what it means. This protein turnover. Let's go to the gym. So protein turnover is here. So this is what happens. Let's say we have protein in the body. That protein is degraded. Generates amino acids. Some of those amino acids are reutilized for a new protein to be synthesized. Some of those amino acids are catabolized. And in this case, when I mean catabolized, I mean, for example, this amino acid is broken into pieces. The amino acid comes apart. Here's what we're talking about. Protein turnover. Have you ever heard that term? That's the dynamic balance between protein breakdown and protein synthesis. This is occurring 24/7. 24 hours a day, 7 days a week, our proteins are being made and being degraded. One of the advantages is that provides a means of adapting to conditions such as diet and stress. Let's say, for example, we do want to lose weight. If we made proteins and kept them for years, we really wouldn't have much adaptation. But what we can do is, let's say, for example, we change our physical regimen when we go from something where we're not exercising at all to something where we're using a lot of our lower body for exercise, we can actually kind of change our body structure, move the proteins around so that now we're providing more muscle on the tissues that are going through stress. We can use this to lose weight if we need to. We can use it to gain weight. So that constant adaptation is advantageous. I'm just going to give you a number here. Estimates of protein turnover. So one of the questions is, how common is this? If a person were to measure this, protein turnover is estimated to be approximately 4.6 grams per kilogram body weight. Let's say a person weighs, that's my example, 70 kilograms. That means in a 24-hour period, approximately 320 grams of their body's protein is being turned over. It was part of a protein 1 minute, it became an amino acid, and now it's going to be part of another protein. 320 grams a day. Is that approximately how much you eat per day? You guys eat 320 grams of protein a day? Nice. How much do you eat a day? What's the RDA for you? Is that the RDA? 70 kilogram person. About 56 grams a day. Sedentary. Now, why isn't our RDA higher? Because we reutilize amino acids. When we take a protein and we degrade it, a lot of those amino acids are used to make a different protein. So in other words, let's say on Monday, I have a protein that's an amino acid that's in my bicep. On Tuesday, it might be a hepatic enzyme. Protein's degraded, goes into the blood. When a protein's made in the liver, it can take that amino acid from the blood that used to be in my arm and use it to make an enzyme. What does that do to our dietary requirements? Increase or decrease? Significantly decreases. That means I don't have to consume 320 grams of protein. Every time a protein's degraded, those amino acids are not lost. Most of them are reutilized. Otherwise, I'd be looking at this instead of this. It's a big difference. So one of the keys to take from this is the most common source of amino acids for protein synthesis are endogenous amino acids. The most common source of amino acids for protein synthesis are endogenous amino acids rather than what? Dietary or exogenous. Yep. Okay? And if you do not stimulate proteins, after a while they tend to degrade. And if you look at something like bed rest, as soon as a person, an older person or whatever reason, about a year and a half ago, a friend of mine was in a traumatic motorcycle accident. The wasting that he was going through was unbelievable when he was in intensive care. That kind of scenario basically just starts degrading proteins at an unbelievable rate, and their body's adapting to that. They're in a complete catabolic phase. A lot of those amino acids are being pulled out. In that case, what happens, a lot of them are being excreted from the body because you don't have anabolic stimuli. So it's very important for people to remain active and use their diet the best they can to maintain that protein. I'm going to stop there. Any questions? Okay, guys. I'm going to I'll load this and I'll let you know when this loads. I'll send you an email. 1 second.
Which is not rather than